everyone, so today I am going to talk about structural insight into the role of prosequence in preproprotein folding and function. So let's understand what is prosequence. Prosequence is additional structure of amino acid which is translated along with the protein. These proteins are termed as preproproteins and the prosequence upon its function is cleaved off. First member to be reported with prosequence is subtlicin in Bacillus subtilis, but further other proteases, neuropeptides as well as growth factors in other organisms were found to have prosequences. So till date there are 2140 prosequence assisting proteins which is present in the database proseq-APBJC, which is formed in our lab by me and my guide. This is found in 690 organisms, and these prosequences were reported to be present mostly at N-terminus, but C-terminus prosequences as well as some of the prosequences in between the preproteins are also found. Here in this study we took only N-terminus and C-terminus prosequences. Further, it was seen that these prosequences were majorly present in the function of attaining active structure of protein. Hence these prosequences are termed as intramolecular chaperone. These prosequences were later found to have other functions like enzyme targeting, signal transduction, as well as preventing the protein from other protease damages, as well as precocious activation of protein. So what do we wanted to do? We wanted to see the involvement of prosequence in modulating protein structure, as well as understanding the residues involved in forming active binding site as well as ligand binding. So how did we attain it? So we took data sets from proseq-APBJC for N-terminus and C-terminus containing prosequences. Then we blast this against the PDB structures, and upon result we took only data sets with 100% sequence coverage as well as 30% sequence similarity. And later on we did residue interaction analysis with for prosequences as well as proteins. Further, we did a pairwise alignment of the protein from the data set as well as PDB structures. Later, we did a Cas-P analysis to find the ligand binding pocket, and further we did the ligand residue analysis through PDB sum. So we found that there were 127 structures of PDB having N-terminus prosequences and 14 with C-terminus prosequences, but there were no further study of prosequence and protein interaction in case of C-terminus. Therefore there is no study further for C-terminus prosequences, but in case of N-terminus there were 31 preproteins with prosequence protein interaction. Further, there were 14 structures at Cas-P, and PDB structures for PDB sum there are 12 structures. So we analyzed that there were possibility of having prosequence directly involved in binding pocket, prosequence interacting with protein residue directly, and prosequence interacting with protein residue which is helping other protein residue to form an structure or binding pocket. So here 1, 2, 3 residues can be seen like that. So we found that there are 12 preproteins with having binding pocket interactions with prosequence directly and indirectly, while there are four preproteins having prosequence involved in binding with the ligand. Mostly there are zinc, calcium sulfate and other ligands which are involved in binding with the prosequences. So if you see here, you can see different number of structures with residue 1, 2 and 3 involvement to the binding pocket as well as ligand. So if we can say that not all prosequences are directly with the cognate protein, but they helps in interacting and forming the binding pocket, and mostly all of them are found at N-terminus. So this was the main highlight, and only few residues of prosequences are in direct contact with the binding pocket, while a smaller portion is actually forming the binding pocket. Residues of prosequences are directly involved in interaction with the ligand as well. So we want to further extend our study to understand the conservation of these binding pocket sites, as well as want to perform like NB simulation to understand the effect of mutation in case of these prosequences. So I would like to thank my guide Professor Manisha Goyal, Head of Department of University of South Campus, other faculty members, seniors, juniors and my non-teaching staff, as well as University of Delhi South Campus, and the funding agency UGC Non-Net, CSIR, SRF Direct, ICMR as well as Delhi University for providing the resources. Thank you.